Hello, my name is Adrian Ely, and this is a video presentation of a small study that was conducted by the EBNet Social Sciences Working Group. That is myself, Josie Coburn and Duncan Moore from Sussex, Jane Calvert, Rob Smith and Kyle Parker from Edinburgh, and Ismael Ràfols, who was at CWTS in the Netherlands when the study was conducted. We were contacted by EBNet in 2023 and had some interesting discussions about the role of social sciences in this BBSRC and EPSRC network, and these discussions led to a short 6-month project that was commissioned as an input from innovation studies and science and technology studies to wider network building in EBNet. We completed the study in early 2025, and this presentation is adapted from one that I gave at the UK Association for Studies in Innovation, Science and Technology conference. That's the UK Assist conference in Edinburgh of that year. The study drew on a novel combination of techniques and research methods to track the changing nature of environmental biotechnology over the last 50 years. We used quantitative bibliometric data from that period, analysing and interpreting it using dedicated software. We also used qualitative documentary analysis, 12 semi-structured interviews with scientists and practitioners in the field. We produced a draft report in late 2024, and this provided an initial mapping of definitions and clusters within the field and identified key issues from interviews that were further discussed at a workshop that EBNet convened in November 2024. We subsequently wrote up aspects of those discussions, and the final report, which is seen here, was published in February 2025. As well as analysing the past and the present of environmental biotechnology, the report also drew upon workshop discussions to present tentative conclusions regarding the field's potential future directions. So first, we wanted to look at the use of the term environmental biotechnology, and we can see from this Google Ngram that its use has increased over time from around 1980 onwards. So the graph draws upon the Google total English corpus from 1930, when use of the term was 0, through to 2022. It's interesting to consider what was going on around 1980 when we see the use of the term environmental biotechnology really picking up. Well, in the USA, Congress passed the Comprehensive Environmental Response Compensation and Liability Act, CERCLA, otherwise known as Superfund, to develop a national program to address hazardous waste dumps. And this created a potential market for some types of environmental biotechnology. A potential market. 1980 was also the year of the famous Diamond versus Chakrabarty case in the USA, the first that upheld a patent on a living organism. In this case, the patent was for microorganisms with applications of relevance to clearing up oil spills. But beyond that, the patent reads, conceivably, plasmids may be discovered that will provide requisite enzyme series for the degradation of environmental pollutants such as insecticides, pesticides, plastics, and other inert compounds. So it's quite interesting to reflect on how the future of the field was envisaged back then. So after this period of increasing use of the term in the 1980s and 90s, peaking in 1994, we then see a reduction towards the early 2000s, and then seemingly a rise again in recent years to 2022. We can speculate on the reasons for this, and obviously this is not necessarily academic literature, it's the total English corpus. But one of the key questions that we wanted to ask was what this term environmental biotechnology really refers to. And to investigate that, we turned to the academic literature and applied bibliometric techniques. The bibliometric study drew upon a corpus from 1973 to 2022 from the Dimensions database. That used a group of search terms which were informed and to some extent pre-framed by consultations and interviews with environmental biotechnologists, including but not limited to those within EBNet. This set of search terms fetched a total of 200,963 publications, which were research articles, review articles, research chapters and conference papers. So these were collectively drawn from 12,745 different sources, that is different journals, edited books, etc. The full process for identifying our search terms, which is always a really key aspect of any bibliometric study, is detailed in the report, and we used this corpus as the basis for all of our bibliometric analysis, which was led by my colleagues Josie Coburn and Duncan Moore. Taken as a whole, it's clear that environmental biotechnology is a growing scientific field, and that growth has continued through to the present day, according to our study, and doesn't show the same ups and downs as we see in its use in general, the Google Ngram corpus. So this doesn't mean, to clarify, this doesn't just mean that there are increasing numbers of papers being published each year in the areas defined by our search terms, but it also means that this number is increasing relative to the number of publications across the whole of science, which are also obviously getting bigger over this period. And it's the increase in the percentage of science that relates to environmental biotechnology that's illustrated in the graph here. That's an aggregate level. We actually found that patterns of publishing activity across the world are actually severely skewed between countries. In the USA and more recently in China and India, these are the countries that publish the most papers on environmental biotechnology. The differences in publication activity between countries are dramatic, and more than an order of magnitude difference in the number of publications between those leading countries that I mentioned before and the 20th most highly publishing country. In order to make these more subtle differences discernible, the map below uses a logarithmic scale to show the publishing activity in each country. White represents 0 publications, and the darkest shading shows the highest number of publications. Different countries also show differences in their levels of international collaboration, and that's indicated by co-authorship, and it's also covered briefly in the full report. So we defined our search terms that we used in the previous 2 slides on the basis of the types of science that were usually associated with environmental biotechnology, according to the scientists that we spoke to. But one of the questions we were most interested in is, to what extent do those conducting these types of science actually use the term environmental biotechnology? And the answer is surprisingly few. Only 881 of the publications in our corpus, which you'll remember is over 200,000, used the term environmental biotechnology in their abstract or title. And this raises questions about the field's nature and its cohesion, at least under this label of environmental biotechnology. One point to mention is that a higher proportion of the text that used the term compared to the full corpus were actually review articles. So this suggests that scientists use the term environmental biotechnology more when they're communicating to each other about the field, rather than when they're reporting scientific findings from within it. Looking across the corpus, we can also see that various related areas, illustrated by terms like environmental engineering, industrial biotechnology, and more recently synthetic biology and engineering biology, also appear in the titles and abstracts. So this illustrates that they overlap with environmental biotechnology. And if we look at the number of papers in our corpus that include the terms environmental engineering, industrial biotechnology, synthetic biology, and engineering biology in those titles and abstracts as a percentage of the number of such papers in the whole of the Dimensions database, we can actually see the extent to which they overlap. And we see a larger overlap between environmental biotechnology and environmental engineering and industrial biotechnology than we do between environmental biotechnology and synthetic biology or engineering biology, which are obviously much newer terms. In fact, the corpus only went up to 2022, which explains why there's a relatively small number in synthetic biology and a very small number in engineering biology. The ones that appear in our corpus is just 2 papers on engineering that mention the term engineering biology, one from the UK and one from the USA. Again, you can see the USA, China and India are leading countries publishing in each of these overlapping areas, and Germany also plays an important role in the industrial biotechnology area. Looking in more depth using these bibliometric techniques, we also asked another question, which was whether it was possible to identify topic clusters within the field. The map shown here is based on what we call bibliographic coupling, where documents, articles, share at least one common reference, and their proximity on the map is based on the number of references that they share. So it gives an idea of how closely related they are. Specifically, this map is based on the top 1,000 documents in the corpus, as they're characterized by total link strength. So it shows the papers that are the most strongly linked. We mapped the field in this way using software called VOS Viewer, which uses an algorithm to enable clusters of related publications to be identified. The clusters are highlighted with different colours, which correlate with various topics, which we had to identify and characterise manually by reading the titles and abstracts. So we have to take care when we're interpreting this data, but the exercise provided an interesting view of 5 possible different clusters. These were identified as general bioremediation, which is in green, contaminants and phytoremediation in grey, biochar, which is shown in orange here, anaerobic digestion shown in blue, and nitrogen and phosphorus waste, which is shown here in yellow. Interestingly, we see relatively little evidence of a cluster mobilizing knowledge from across all of the various parts of the network. The different clusters seem to have developed quite independently within our corpus, and there's no cluster that uses knowledge from across the whole field. The way that this was interpreted at the workshop was that environmental biotechnology is somewhat like a donut, so it's made up of different subfields around the outside without a recognizable core at its center. As the field has developed over the last 50 years, publishing activity across these clusters has also changed. So we've seen more publications in recent years at the peripheries in the biochar, anaerobic digestion clusters. We also created maps that show different specializations across these clusters and in different countries over time, and you can find some of those maps in the report. We also provide a description of the longer history of what we might understand as environmental biotechnology, going back to ancient times, but with increasing understanding of microbial activity being associated with what we call later generations of the field of environmental biotechnology. The later generations see a shift from dealing with issues of urbanization, characterized by areas like public works for wastewater management and sanitation, towards cleanup of wider problems of modernization, and especially environmental problems caused by new technologies, chemical technologies often. But how about current dynamics? How is the field changing now? Well, we investigated this through our interviews, and we found that several of our interviewees pointed towards an emerging generation of environmental biotechnology deploying tools from synthetic biology or engineering biology. This they claimed draws upon capabilities not only in biology but also in engineering and increasingly in digital technologies. But the crucial difference is that the emerging activities in the field relate to the manipulation of microorganisms at the genetic level rather than, or as well as, at the level of their communities. And this points to a number of interesting questions, some of which we list here. I've already discussed how biotechnological aspects of environmental biotech, for example, microorganisms, became patentable or become more patentable since the 1980s. Patenting genes has also been enabled by constructing them as a chemical compound. So one question is whether this shift towards the new generation of environmental biotechnology around genomes means an increase in proprietary technology at the expense of public goods, or what used to be characterized by public works. As we progress to the new generation, is that rise in patented proprietary technology inevitable? There's also interesting questions about the role and claims being made for environmental biotechnology, and at least the ambitions claim to respond less to the environment from a local level, so waste management, for example, and more towards environmental challenges, the scope of which can be seen as at the global level. So, for example, there's been more recently an increased focus on biochar, microbial fuel cells, as shown in the bibliometric study, which could be seen as a response to climate change. But there are also environmental biotechnologies that are being put forward to address other planetary boundaries, so environmental challenges at a global scale. Some interviewees pointed towards a greater role for environmental release and learning by doing, so learning from experiments in the wild, rather than laboratory-based experimentation, which currently characterizes synthetic biology and engineering biology. And our interviewees pointed to opportunities in this sense, but also obviously risks and governance challenges. And we did take the opportunity to discuss some of those with them in the workshop. So at the workshop in November 2024, we talked about some of these challenges and also posed questions for the future. And these are laid out in much more detail in the report, but very briefly they included the following. Firstly, what might environmental biotechnology as a field look like from other perspectives, with the corpus defined differently? You know, if we spoke to different scientists at the beginning, they may have given us somewhat different search terms. For example, we haven't looked at environmental DNA, we haven't looked at technologies such as gene drives for biodiversity conservation, but if those had been included, that could have given us quite a different perspective on what this field looks like. Also, how would the overlaps with the other related areas look if we started from a corpus that defined, for example, synthetic biology, which other studies have done, but without really looking at the link with environmental biotechnology. Secondly, some interesting questions about patenting, something that came up in our discussions. And so we think it's quite interesting to look at, it would be interesting to look at the evidence around patenting, to investigate how intellectual property is contributing to or hindering innovation in environmental biotechnology, and to look in much more depth at where and how environmental biotechnologies of different kinds are being used on the ground, and what role IP plays in those different applications. Another one of the governance challenges that were identified at the workshop related to claims that environmental biotechnology could potentially convert increasing numbers of waste streams into useful industrial inputs, thus fostering circularity in our economies, circular resource flows and circular economies. But in doing so, some participants suggested that they might actually sustain demand for those same waste streams, and in so doing make it harder to disrupt the damaging industries or systems that actually produce that waste. So one of the emerging areas which was seen as really important was to examine how emerging environmental biotechnologies articulate with current unsustainable socio-technical systems, and to ask how policies can ensure that these technologies foster a transition away from unsustainable approaches rather than actually contributing to further lock-in to wasteful or polluting industries. This was just one among many questions raised about policy and regulation and how they can adapt to this rapidly changing field. A key concern in this regard was what kinds of regulatory frameworks would be proportionate across different applications of environmental biotechnology, with different risks and uncertainties, whether those are known and well characterized or unknown. How do the regulatory frameworks differ across different contexts? They have to be context specific and adapted to the socio-political, environmental and wider social conditions in which they're being implemented, and also how can these different regulatory frameworks best be coordinated at the international level as we see environmental biotechnology being taken up across a more diverse and broader range of countries across the world. So those are just some of the questions that were asked, discussed at the workshop, and also reported in the main report. The report is available in full as well as in executive summary version at ebnet.ac.uk. And if you're interested in the work and possible future directions listed above, feel free to contact me on the email address here. Thank you very much.